Well, welcome everyone to this little podcast. I thought it would be a good idea to spend a little bit of time before class just talking about DNA replication. This can be a kind of a complicated process, particularly when we start talking about some of the proteins that are involved in the process of replicating DNA. So for the most part, what I'm going to do is focus on the information on Objective 2 on your study guide. All right, so as we begin looking at DNA replication, we see that DNA is replicated in a mode that is called semi-conservative. And so what does that mean? Well, let me just draw a piece of DNA here. And I'm not going to draw a double strand. I'm just going to draw it as a link here. Now, semi-conservative, what that means is as this DNA is replicated, it's literally ripped apart, and you have one strand over here and one strand over here. And then when the new DNA is synthesized, it's laid down right next to the original piece of DNA. I'm just going to draw that with a dashed line, just to symbolize that it's a new piece of DNA. And so the reason we call this semi-conservative is because semi for half and conservative for maintaining the original structure you have. So we're able to maintain, each new DNA molecule maintains half of the original. So that's why we call it semi-conservative. I'll say that again. Each new DNA molecule that is produced conserves half of the original piece of DNA. So this is replication. All right, now let's look at replication a little bit more closely at the molecular level. Okay. So here's our DNA molecule. I'm drawing it the other orientation now. And it opens up like so to begin to make new DNA. And it opens up like so in such a way that we call this the replication fork. So I'm going to, let's see, this program doesn't allow me to write very well, but this is our replication fork. Right here. This whole thing is called the replication fork. I guess I could put REP in front of it just to remind us what we're looking at here. Bacteria's chromosomes are circular. And so when it begins to make DNA, it just has one replication fork. Well, actually two, one going each way. But there's only one origin of replication right there. Okay, now in eukaryotes, our chromosomes are so much larger, and I'll just draw something like this. They're immensely bigger than what the bacterial chromosomes are, even though this picture doesn't really justify the size differences. But because of that, in order to replicate all the DNA in a timely manner, there are multiple origins of replication in eukaryotes, particularly organisms like humans. Even though there are differences, such as the differences of origins of replications, it's important to recognize that the process of replication is the same in bacteria versus eukaryotes. There's very few differences. It uses the same kinds of proteins, the same kinds of enzymes. So let's talk about some of these enzymes real quick then. We're only going to talk about a few of them, but recognize there are many more than what we're going to talk about here in this chapter. You'll get much more of this as you move into cell biology and genetics. The first enzyme we're going to talk about is helicase. And what helicase does, and we're just going to plop it right here at the opening of the fork. And we're just going to put an H here, but that's for helicase. It sort of acts like a buzz saw. It moves down the double helix. And remember, I haven't drawn them here yet, but let's go ahead and draw some hydrogen bonds. So the hydrogen bonds between the complementary bases. You should know what the complementary bases are from your reading. Between the A's, G's, C's, and T's. Okay. Now what helicase will do is that it will rip down through the double strands of the double-stranded helix and break all of these hydrogen bonds, rips them apart like a buzzsaw. And it does so in such a way to open up the DNA fork, exposing the single-stranded copies of the DNA molecule. Now, a couple other enzymes come into play here that we should talk about. The first are single-stranded binding proteins. Single-stranded binding proteins, what they will do is they will bind to the single strands of DNA. It's probably the only enzyme we will ever encounter that has the perfect name that describes its function. So single-stranded binding proteins. And that's literally what they do. They bind single-stranded pieces of DNA, and they do so to prevent these two strands from coming back together. Single strands of DNA naturally want to come right back together, but if you can clog them up with these proteins here, it'll prevent these strands of DNA to come back together. Remember, we want to keep them separated so that we can make new DNA copies using this strand here as our template. Remember, the semi-conservative model says that we're going to conserve half of our DNA molecules. All right, now let's talk about another enzyme called topoisomerases. Your book calls them topoisomerases, and that's the eukaryotic version of it. But in bacteria, what it's called are gyrases. We'll stick with topoisomerases, but you should know that there's a distinction between a topoisomerase and gyrases. Okay. So a topoisomerase, what it will do is it'll bind the DNA, and what happens here is as the helicase is breaking these bonds of these hydrogen bonds, the DNA strand will begin to kink up. If you were to take a cord and hold it tight at one end and spin it around and then begin to pull it apart, maybe we'll do that in class just to kind of show you what I'm talking about. What happens is that this DNA molecule will begin to stretch and eventually it'll break. What the topoisomerases do, let me put a T here so we don't forget that this is topoisomerases, it will literally cut the DNA and allow the helicase to unwind it without putting any extra stress on the DNA molecules themselves. So what they essentially do is they keep them from breaking. Okay, and a quick little side note here, as I said, in eukaryotes it's a topoisomerase. So let's just write T for topoisomerase, and that's what we find in eukaryotes. Now bacteria, they use an enzyme that does the exact same job, but it's a little different, and it's called a gyrase. I'll go ahead and spell that one because it's not in your book. It's Gyrase. And this is what prokaryotes use. And the reason I want to mention this is to offer an example of why it's really important for us to learn the basic biology of bacteria, of eukaryotes, because understanding a topoisomerase and gyrase has led to a very effective antibiotic drug. There's a drug that you can get, and what it does is if you have a bacterial infection, you'll take this drug, and it'll go after the bacteria only because it goes after the gyrase. It makes it so that the prokaryote can no longer relieve that torsional stress. So every time the bacteria tries to divide, it breaks the DNA apart and it can't divide. It doesn't, you might worry that this would affect human DNA polymerases, human topoisomerases, but it turns out it doesn't. The topoisomerases are unaffected by this particular antibiotic. All right, now there are three more enzymes we need to talk about. The first is called a primase. And what primases do is they lay a little piece of nucleotides. It turns out it's RNA, but that's not important for right now. This is RNA that's laid down. And the reason that this is important is because, well, let's back up for 1 second. This RNA primase, let me put a P here, and let me back up 1 second. This is an RNA primer, and I just lay down the word primer here. So it lays down this primer. And the reason it does this, let me back up 1 more second. It does so by using an enzyme called primase. And let's just go ahead and put a circle here with a little P here for primase. That's the name of the enzyme that lays this primer down. Now the reason it does that is because another enzyme called DNA polymerase will bind right here. But it can't bind to DNA by itself. It's unable to do that. So it has to bind to this RNA primer. When it binds to this RNA primer, it can begin to synthesize DNA. In this particular side, it's going to go in this direction. So as the helicase opens up the strands and makes single-stranded pieces of DNA, the DNA polymerase, and I'm just going to write D polymerase here, so we remember that that's the DNA polymerase, it will move along here adding DNA, and I'm going to show the new DNA as being black because I'm almost out of different colors. The end product of what we're drawing here is going to look something like this. And I'm just going to show the top strand right now. So here's the new strand that was just formed, and here's the original strand. Okay. This is our finished product. Okay, if you notice up here, as I drew the the DNA polymerase, and it was moving towards the replication fork, and it made DNA going in this one direction. It's doing this in part because of the polarity of the DNA. And so let me go ahead and write the word 3' here and 5' here. The enzyme reads the DNA in a 3' to 5' direction. Okay. The new DNA that's being made is the reverse, because one of the rules of DNA is that it's always anti-parallel. So the new DNA is 3' and on one end and 5' here. I'll just scribble that in here. Okay, now because of this, what happens is this other parental strand here has to be in the opposite direction. It has to go 3' to 5'. As so. So that creates a slight problem because it still reads it in a 3' to 5' direction. So what happens is that this piece of DNA laid the primer down and then made new DNA through here. And then the fork opens up a little bit more and you put a primer here and it lays new DNA down like so. And every time the fork opens up, it has to lay down a new primer with the primase, and then the DNA polymerase adds more DNA. And the result of that is, and let's just draw this down here, something we call, well, there are these little fragments. The black, remember, is the new DNA that's just been laid down, and the red is the old piece of DNA, the original piece of DNA. And so what we call these are Okazaki fragments. I'm just going to put OF for Okazaki fragments. So our end product are 2 new pieces of DNA. However, this piece of DNA down here, we have these big gaps in them. And we can't have those gaps because that would not be sustainable for the DNA. So our final enzyme that we're going to talk about, we're finally there. And I think I'm officially out of new colors. I take that back. I still have the color gray. So we'll use gray here. This enzyme is called DNA ligase. DL for DNA ligase. And what it does, so DNA ligase, it will bind to the DNA, shown here, and fill in these gaps. It'll fill in each of these gaps between each of the Okazaki fragments. So the end result now is 2 fully formed DNA molecules that will then get packaged into a new cell when the cell divides. Okay, so that's it for DNA replication, essentially objective 2. What I would recommend as you're learning this and all of the different enzymes involved and all the different structures involved, I recognize it can be quite complicated. I would, on your own sheet of paper or find a whiteboard somewhere, draw this out. Draw it out many times. Draw the different enzymes in their place. Write down what they do. Maybe flashcards would help. But you will definitely have to know this information. So draw pictures and come see me if it's still unclear. Some of the questions I like to ask with this kind of a molecular nature is I might ask you questions, might even be draw this, or it might be identify the different proteins. Like I might draw it on a question and then ask you to identify the different proteins involved. All right, well, good luck going over this material. Please let me know if you have any questions, and I'll see you in class. Bye.